Human research is the ideal kind of research to do because we are concerned about effects in humans. But it's very expensive to do human research. As you can see from this Venn diagram, let's go through the different kinds of human research. If you want something tightly controlled with a high subject number, it's going to be pretty short duration. If you want a longer duration with a high subject number, it's going to be low control, i.e. free living. If you want a tightly controlled study with a longer duration, it's going to be a low subject number. And if you want long duration, tightly controlled, and high subject number, you'll have to do it in animals. These limitations of research are part of the reasons why one line of data or a single study should not be sufficient to move the weight of scientific consensus. So let's go through the hierarchy of evidence. At the bottom here of this pyramid, we have editorials and expert opinions. We should value the opinions of experts, but they are still just opinions, and those opinions are clouded by bias and personal beliefs. Next up, we have mechanistic studies. We're going to talk more about mechanisms later, but these studies tend to be done in vitro or petri dishes or in animals. And when we talk about mechanisms, we're typically looking at a single biochemical pathway and looking whether or not a particular treatment impacts that pathway. We're not looking at a hard outcome like, say, fat loss or lean body mass accrual. We're looking at, say, phosphorylation of mTOR in response to a specific treatment. Then we have case reports and case studies in humans. Again, reasonable to look at, but can be unreliable. You can find case studies where people did something and then something happened and they are assuming that the two things are related. But the two things may not be related. For example, every morning I have breakfast and then I go type on my keyboard and answer emails. One might conclude, based on a case study, that eating breakfast causes me to type on my keyboard, but that would be an inaccurate conclusion. Furthermore, this is only one data point in a single person with no actual intervention. Then we have cross-sectional studies or surveys. This is where they look at different sections of populations and attempt to correlate some variable with the differences observed in those populations. Let's use a made-up example. Maybe people in France have lower rates of heart disease than people in the United States. And perhaps they've been able to correlate that to olive oil intake. People in France eat more olive oil, people in the United States eat less olive oil. That doesn't mean that there's a causation there, because people don't just eat one type of food. People's lifestyles tend to be wrapped up together, and people with better dietary habits tend to live healthier overall lives in general, whereas people who eat, say, more processed foods tend to have overall unhealthier lifestyles. And it's very difficult to tease these things apart. But these surveys or cross-sectional studies can be helpful to identify questions that should be asked and then try to answer with more tightly controlled studies. Then we have case control studies. These are where an intervention is attempted to be applied to a person or series of people and then look at the outcomes. It's typically low subject number with not as tightly controlled as a randomized control trial. Then we have cohort studies. Cohort studies are still epidemiology because you're looking at populations that are different, but the nice thing about cohort studies is they are done over a period of time. So for example, instead of doing a cross-sectional survey where you're looking at one population versus another as a snapshot in time, in a cohort study, you look at different groups of people over a period of time, say 10 years. And you look at the incidence of disease or some outcome. So for example, you might look at people who follow a low carbohydrate diet versus a high carbohydrate diet over 10 years and look at who has more heart attacks over that 10-year period. The advantage to this kind of study is each person is acting as their own control over time. The downside of this study is it's still very difficult to disentangle people's overall lifestyle from a single variable. And as you can see here on this chart, as we go up in the quality of evidence, the risk of bias becomes lower and lower. That brings us to randomized control trials, which are considered the gold standard for research. When we say randomized, what does that mean and why are we randomizing? So if we have two groups of people and we let them, for example, self-select what treatment the group they want to be in, there is a high probability that people with certain characteristics are more likely to select a certain treatment versus other people. This means there is still a high risk of bias. However, if we randomize them, which means you put them in groups randomly based on a certain outcome. So for example, if body weight is the outcome we want to look at, our primary outcome, we'll randomize people based on body weights. So for example, if we have 100 people and the average body weight is 75 kilograms, we will randomize people to different groups, but we'll try to make sure that overall the average body weight between the two groups is not different. This removes a lot of the bias because people are not self-selecting, and it means that if there's any difference between the groups, it's more likely to be from the treatment than some sort of inherent characteristic of the subjects themselves. Then finally, at the highest level of evidence, we have meta-analyses and systematic reviews. Now you can do meta-analyses and systematic reviews of cohorts and also randomized control trials. A meta-analysis is basically a study of studies. What they're trying to do is put together multiple studies based on specific inclusion criteria. So for example, if they want to look at low carbohydrate versus high carbohydrate diets, they might use inclusion criteria such as the subjects have to be non-obese, non-type 2 diabetic, and have no pre-existing health conditions. They have to be active but not engaging in more than, say, 4 hours a week of exercise. And the studies had to equate calories between the diets. I'm just giving some examples of what inclusion criteria could be. But the researchers will then take this inclusion criteria and include every study in their analysis that fits this inclusion criteria and try to come to an overall consensus of the data. What do all these studies, when summed up, show? So in that way, meta-analyses and systematic reviews are considered the highest form of evidence. However, I don't want you guys to become dogmatic about this pyramid. Certainly, there are some really well done cohort studies that I would value over some poorly done randomized control trials. I've seen meta-analyses that have poor inclusion criteria for the conclusions that were drawn. But in general, this pyramid holds true that as we reduce the risk of bias, the quality of the information and the quality of the data improves.